all kinds of different names for the same sort of things. RYRs are referred to as adenine receptors, largely because of the plant alkaloid that blocks the calcium release units. They're all the same thing. And the dihydropyridine receptors, again, is a drug that blocks these channels, and they're basically L-type calcium channels. And I don't imagine that Dr. Norton spent any time talking about calcium channels, but they become really important in all kinds of aspects in cardiac muscle and also skeletal muscle. Okay, so those are the same things. If you read a textbook where I sort of talk about calcium release units, I'm actually talking about adenine receptors or vice versa. And these names are basically based on the pharmacological agents that inhibit or block. Okay, so we finished off last day with this point.
I think he said they
Calcium
could block
the L-type
because
calcium channels
they can
is an
just
absolute
we block calcium channels.
requirement
That's what I think.
for cardiac muscle to contract. And here's some, tells you something about the difference in concentrations, not much inside, and about 1.8 to 2 millimolar calcium outside the cell. This is not the case in skeletal muscle. Skeletal muscle, although L-type calcium channels are involved, you don't need calcium outside the cell. Cardiac muscle is an absolute requirement. And as I said, on the sarcoplasmic reticulum has calcium release units or channels. They're called adenine receptors or RYRs or what have you. This is a schematic representation of the organization between transverse tubular system and cardiac muscle. Also applies to some degree skeletal muscle. This is the terminal end of the endoplasmic reticulum. It's called a terminal cisternae. That's just the little sort of bulbous ends that you saw on the diagram of skeletal muscle. Inside the terminal cisternae is something called calsequestrin, and its function is to actually bind free calcium. So there's a lot of free calcium inside you bound with this protein. Also shows the calcium ATPases or the circuit pumps, which when the calcium concentration outside goes high, starts to work at the expense of ATP and pumps calcium back into there, into the terminal cisternae of the SR. This is the bilayer membrane of the transverse tubules, and on there, I guess, there are proteins which are actually L-type calcium channels. This is artistic representation. On the surface of the terminal cisternae are these sort of foot-like processes, which are in fact the actual RYRs or their adenine receptors or calcium release units. So they're in very close opposition. So basically, if the cardiac muscle, if this is depolarized, the L-type calcium channels open and they allow calcium to enter this region here and activates the adenine receptors of the calcium release units. The mechanism by which calcium binding in cardiac myocytes to these calcium release units and how this causes a release of the bound calcium is really unknown at the moment. But anyway, if you get calcium binding to somehow or other calcium, there's probably some sort of charge transfer, releases a ton of calcium from these guys here. This calcium release units which are located here. Another look at this. Sort of very that are colored is in fact another one to look at this is in fact the transverse tubule, large cardiac myocytes. This is the endoplasmic reticulum of the terminal cisternae. are the ryanodine receptors of the calcium release units, and the L-type calcium channels are right there. There's also calcium channels all located pretty well all over the membrane. Now that's what increases calcium concentration, so the action potential travels down here, depolarizes this membrane, activates these L-type calcium channels. Calcium goes into the cell and binds to these receptors, and you get a release of calcium from the SR to cause contraction in the cardiac myocytes. We're going to go over this when we're talking about cardiac muscle. And of course, there has to be pumps to remove the calcium to cause the myocyte to relax. And that's accomplished by this calcium pump primarily at the expense of ATP. In cardiac myocytes, there's of course the exchange mechanism. We're going to talk about that in the next series of lectures. And of course, the standard pumps that generate or are important in generating, I'm talking about this one, membrane potential. These are exchange proteins, and this, of course, most cells have an ATP-driven calcium pump in the plasma membrane. It's a high affinity, low capacity one, and it actually removes some of the calcium to the outside. But the important one for this series of stuff is this one here, which removes the calcium from the cell. And there's in the sarcoplasm particularly, there are tons of these guys. And in heart, big hog for ATP, so it requires a lot of ATP. And of course, mitochondria might be involved, but we won't bother talking about that. Small amount. So the excitation contraction coupling mechanism in cardiac myocyte is called calcium-induced calcium release. So you've got to have calcium outside because it requires external calcium. Mechanism involves the amplification. I'm going to talk about that when we talk about the heart, which enters the cell via the L-type calcium channel, causing a large increase in intracellular calcium. And the internal sort of change in calcium concentration is proportional to the amount of calcium that goes into the L-type calcium channels. So you can modulate or regulate how strongly cardiac myocytes contract by determining how much calcium goes in by these guys. So it's a modulatory thing or mechanism, and that's kind of important for the heart. It's not the case in skeletal muscle, but in the heart that becomes kind of an important issue, and we'll talk about that again a couple lectures down the road. So if you're talking about proportion of calcium, the normal heart when you're sitting there writing these notes and things, you hope this isn't on the exam. It will be. So about 10% of the calcium required for contraction in cardiac muscle in ventricle comes from the L-type calcium channels. The other 90% comes from the sarcoplasmic reticulum. So the stuff coming into those channels is simply a trigger, primarily a trigger. It can be involved, but it is a trigger. Relaxation is covered. It's caused by several mechanisms. I must have froze there last night. I can't remember. Most not working very well. And in the sodium calcium exchanger, we're going to talk about that again. That's kind of important in various drug effects. And the ATP-driven calcium pump in the SR contributes to about 90% in ventricle of the calcium taken back up. So it's roughly proportional of where it comes from and how it's taken back up. So it comes from outside the membranes, about 10%. It comes from the SRs most. This is in ventricle. In atria, or the other chambers, it's about 50-50. And the other thing that we'll talk about the next time around in cardiac muscle, the sarcoplasmic reticulum ATP-driven pump is regulated by another protein called phospholamine. And that regulates how fast the pump takes up the calcium. It regulates the rate of calcium transfer or translocation. And it's regulated by a second messenger called cyclic AMP. Anybody heard of cyclic AMP in this class? And Dr. Barton, I hope? Good. So, cyclic AMP becomes important, and it can regulate the speed at which this pump can pick up calcium. And we'll talk about that when we talk about regulation of heart rate, all that sort of stuff. Now, the other interesting thing about cardiac muscle, in comparison to skeletal muscle, is the fact this length tension diagram, or the optimal length, is totally skewed. It's different. This is the one I showed you last time with all those fancy little actin-myosin overlap diagrams. This is skeletal muscle. It has a fairly large length working range. So it goes from, you generate from about 1.4 micrometers to about 2.8. However, cardiac myocytes have a very sharp length tension diagram. And most of the action in the heart is roughly in this range here. It's on the ascending limb. So that means that the actins are kind of bumping into each other. If you stretch it over this length here, which is about here, about 2.4 micrometers, the tension that the myocyte or the cardiac muscle can develop will drop off substantially. And in the entire heart, this would be a region where you would have what's called heart failure. Can't generate tension. And if you stretch it in this region, the other thing that happens is you actually strip or rip off the tightened filaments or tightened muscle protein. So there is no elastic recoil. So at these lengths, all the tight and all the good stuff that tighten supposedly does disappears. But your muscle in your heart works roughly in this range here. So it's totally different than skeletal muscle. And just to sort of summarize, short sarcomere length, the fall and tension is due to a couple other factors. The reduction of this sort of thing, affinity for the troponin C, or the calcium binding protein, and a reduction in the amount of calcium that goes in. At sarcomere length greater than 2.4 micrometers, active tension declines, and the elastic recoil of the plate impedes the development of active tension. If you go too far, you strip off the tightened filaments and no longer provide the elastic recoil. So this is a couple of points. And that's all it for cardiac muscle. We'll be going over some of this stuff again as we talk about the entire heart. We'll just do a bit of stuff on skeletal muscle. Okay, now Dr. Barton has told you a little bit about smooth muscle, because smooth muscle is driven in many cases by the two divisions of the autonomic nervous system. I have some feelings for smooth muscle. Smooth muscle cells are spindle shaped. And they vary in blood vessels, all your sort of arterial blood vessels, for example, which isn't very large. And they can grow in size to about 500 micrometers, which is about a half a millimeter, in the walls of the uterus during pregnancy, so they can change shape as well. They're called smooth because they don't have any banding or striations as you have in skeletal and cardiac muscles. So they're called smooth muscles. They don't see any striations. Actin and myosin are present, but the relationship is not well organized at all. Actin filaments are oriented mainly parallel or oblique to the long axis of the cell, and multiple actin filaments join in sort of regions called dense bodies, which are found immediately beneath the cell membrane as well as the interior of the cell. And thick filaments, which are made up of myosin, are interspersed somewhere or other along thin filaments. So the totally different structure is no banding, small cells. This is a picture showing you the dense bodies, some inside, some outside. This is one smooth muscle cell. And these little guys here are actin filaments. And this guy here is a myosin filament. It's a different subunit or isomer of myosin. And this is sort of a blown-up version of the whole thing. This is the myosin, these are the actin filaments. So in actual fact, when smooth muscles contract, it becomes fairly, it looks like a spherical muscle. So if you isolate the smooth muscle from blood vessels or something, a contraction, they become, when you activate them, they become like favorite basketballs, very tiny basketballs or baseballs or whatever. So that's their contraction mechanism. Smooth muscle contains most of the contractile proteins: actin, myosin, tropomyosin, something called calmodulin, and an enzyme known as myosin light chain kinase. In addition, there's another enzyme called myosin light chain phosphatase, and that's important in termination contraction. So there's differences. In comparing this to skeletal muscle, what's missing? Troponin, absolutely. Good answer. All right, so that's what's in there. Functional organization, depends where it is. Two basic types of organization. There's something called multi-unit smooth muscle. Cells are innervated by the autonomic nervous system. The neuron makes multiple contacts on the cell, and it actually expands to form what is called the varicosities. What do varicosities do? Dr. Barton's lecture. What's in the varicosities? Neurotransmitter, absolutely. So there's no specialization. It's like the old bucket approach. You just slosh some transmitter over the membrane. And the smooth muscle cells, they have receptors for all kinds of things over the entire surface. Not like skeletal muscle, which is only sensitive to acetylcholine at the end plate. So there's no specialization. No gap junction, but in some cases, in this type of organization, there may be a few gap junctions. The gap junctions are, of course, electrical synapses, right? CNS. So in general, each muscle cell acts independently of each other. Not many of them around. Ciliary body of the eye, piloerector muscle of the skin, some blood vessels. That's the ones that give you goosebumps these days. When you're cold, frightened. Okay, that's the multi-unit. And then the other one's the unitary type of smooth muscle, innervated by the autonomic nervous system, have the same varicosities. Cells are electrically coupled by a gap junction, which allows contraction in a single unit or in sheets. Location, gastrointestinal tract. uterus, bladder, and some blood vessels. So those are locations, different kind of structure. Some contract more or less independently, others contract as sheets or totally as a combination of many cells. Here are sort of pictures of the two organizations. This is the multi-unit one, electrical isolation, fine and control, as you would expect, and the unitary one, of course, are coupled by gap junction. Want to contract a sheet, for example, the bladder. These pictures are beautiful, you know that these guys are trying to contract. They tell you, give you send you a message. In the gut, you get these sort of peristaltic movements, or movements which are oscillatory, oscillatory, and that involves some strange electrophysiology, not strange actually, but it involves two types of channels. One is my old friend, the outside calcium channel, and another one is a potassium channel. Now, I don't know if Dr. Moyne ever mentioned it, but it's a calcium-activated potassium channel. So as the calcium concentration near to the inside of the cell goes up a little bit, it activates these channels, and they pass potassium, primarily potassium. So it's called a calcium-activated potassium channel. So basically, the thing oscillates, the memory oscillates, and if you look at the tension would increase in the muscle in these regions here. So basically, when you get to this stage here, the outside calcium channel is open, and they give you calcium-based action potentials. They're not like sodium action potential, slow, because the outside calcium channels are a slow kind of channel, very slow. Not very slow, but slow compared to sodium channels, which we're all very familiar with. So it goes up, depolarized, you get this sort of hair on top of the electrical activity. And then basically, as you increase the calcium inside the cell, you increase the calcium concentration and the open calcium-dependent potassium channels, and you getting potassium leaving the cell. What should happen to the membrane potential? It's nervous. So if you activate potassium channels of any kind, what happens to the membrane potential? Good point. Hyperpolarized, becomes more negative. So this goes on for a while. You get a slow hyperpolarization. Eventually, these calcium channels will close because the membrane is hyperpolarized. They have to be depolarized to activate them, like sodium channels. And you go back to the stage again where you kind of get these sort of small action potentials. And the size of these action potentials will determine how much the contraction. So this is a gut muscle. It oscillates, and it's all done through muscles through two specific channels. One is the one you've heard of, the outside calcium channel, and it's the one that I surprised you with, is the calcium-activated potassium channel. Now, in the central nervous system, all of these channels actually exist, but you may have not heard. So that's why it oscillates, and it does that in order to move food or whatever along the GI tract, so keep it peristaltic. So you get oscillatory contraction. Now, cross-bridge cycling smooth muscle is again kind of different. You need calcium. An increase in calcium initiates a chain of events that increase ATP activity of the myosin filament. Now, you remember from skeletal muscle I showed you two light chains? And one of these light chains I said could control the ATPase activity of the myosin. So if you increase it, you may want to bind to act more freely or more easily. So the myosin has the same kind of light chains. And what happens when you elevate calcium, you get more calcium ion to another protein, not component C, but to calmodulin. So calmodulin in this case, in smooth muscle, all kinds of smooth muscle, is your calcium sensor. Calcium calmodulin activates an enzyme known as myosin light chain kinase, abbreviated MLCK, and MLCK phosphorylates a regulatory light chain on the myosin head. This increases the ATPase activity and allows it to bind to actin. So it's a different mechanism. Different sort of players, different mechanism. I have a cartoon here, it's stolen from some textbook. Roughly what it is, so we describe it a little bit. You elevate calcium, or you bind to calmodulin, that activates the myosin light chain kinase, and then that phosphorylates regulatory light chain, and then that will allow it to bind to the actin filament that's there somewhere. So that's the whole sequence of events, and it's different. The calcium sensor being calmodulin rather than component C. Different mechanism. So there are all kinds of things in smooth muscle because it has receptors for everything, and many mechanisms which cause smooth muscle to contract. Other mechanism act on the actin cones to remove the inhibitions. Something called caltonin and caldesmin. I don't bother with that. The relaxation of smooth muscle. You now contracts, you want it to relax. You drag your 2 pitchers of beer. Your bladder gave you the message. Bring to the bathroom. Your muscle has to relax. Not only reduction of calcium, but that you also need dephosphorylation of the myosin light chain. That's accomplished by the myosin light chain phosphatase. This guy is not calcium dependent. It dephosphorylates myosin light chain at very low calcium counts. So it doesn't really matter if the calcium high or low. It's there all the time, and it's sort of antagonistic to the phosphorylation. So if the calcium goes low, the other one is not activated, and this causes the relaxation. So it's a different mechanism, but you also need, somewhere along the line, you need a reduction in calcium. Cause complete relaxation. Okay, this is sort of a very complex situation because excitation, contraction, and smooth muscle, the calcium can come in through many mechanisms. Extracellular ligands trigger influx, ligand-mediated channels of various kinds, which may be sensitive, for example, noradrenaline or acetylcholine, and G-protein coupled receptors through IP3. In addition, there's my old friend, the L-type calcium channel, this guy here, and they activate reactivity receptors, and also in addition there's other kinds of things called store-operated channels, depends on the amount of calcium inside the endoplasmic reticulum in smooth muscle cells. These are very poorly understood, except that when calcium drops, it opens up calcium. These guys here are various kinds of channels that allow calcium to go into the cell to replenish the stores. This is a complicated figure, but it tells you, for example, you don't have T system in smooth muscle. No T system, but you have alveoli. These have L-type calcium channels in them. Sort of little indentations in the smooth muscle cell membrane, no T system. So that activates these calcium channels, which actually activates the reactivity receptors in the endoplasmic reticulum. So that's one source. There's a store-operated channel. And of course, there's the receptor type stuff, which could be noradrenaline, could be acetylcholine, could be many other things, which activate a cascade and release calcium from the sarcoplasmic reticulum through IP3 receptor activation. So the whole thing is much more complex than skeletal muscle or cardiac muscle. Although cardiac muscle does have these kinds of receptors. Skeletal muscle is not a big player. But smooth muscle, many ways of getting calcium into the cell. So basically, this is sort of a summary of how these things work. Myosin light chain kinase, phosphorylase, sitting around there very close is the phosphatase, and it's sort of a relaxation. One causes contraction, and the other one does the relaxation. And it's always working. This guy is always working, because it doesn't matter, not calcium dependent. This guy is myosin light chain kinase. So this guy is phosphorylase contraction, but this guy starts to dephosphorylate the actual myosin here. So that's the mechanism. This guy requires calcium, this guy does not. So that's a relaxation process. And in addition, you want to reduce calcium. There's the stuff there that you actually calcium pump, sodium calcium exchange, we talked about cardiac muscle. You have calcium pumps in the sarcoplasmic reticulum, and so there's many mechanisms to reduce calcium in smooth muscle. In much more complex than skeletal or cardiac. The other thing about smooth muscle is kind of interesting. Something called the latch bridge mechanism. And in this case, this generates tension at low energy requirements. It doesn't require ATP and an awful great deal as you would with normal contraction. It's called the latch bridge mechanism, and it remains tension, maintain tension. I hate winter. Maintain tension at very low energy expenditure. It's called the latch bridge mechanism. And I made a mistake this year when I said the term papers. I should have asked people to look it up and read it, because as far as I know, the mechanism is unclear. So maybe you could clear it up for me. Anyway, produces a sustained contraction with little or no expenditure of energy. So once it contracts, it stays contracted, and it's not using ATP to phosphorylate anything. And basically, it's thought that it's something along the lines of the Viger complex. The actual mechanism why the actin and myosin stay attached is unclear. So maybe next year they'll try to see if we clear it up for them. So I can't find anything that makes sense. Anyway, it maintains contraction with little or no expenditure of energy, and it's thought to be something like the Viger complex in skeletal muscle. The actin and myosin are bound and they don't release. Maybe not enough ATP around, but it's similar to the situation in skeletal muscle when you sort of semi-depleted of ATP. So, in summary, I guess, smooth muscle cell, a wide variety of neurotransmitter hormone receptors. A lot of things can contract smooth muscle. You know this guy here, adrenaline, noradrenaline, angiotensin, neuropeptide Y, nitric oxide, something called endothelin, which is a big player in blood vessels, and of course your old friend acetylcholine. So a lot of things have receptors completely already. And a lot of these things are called contract, independent of whether the autonomic nervous system is activated or not. So a lot of receptors. Cardiac myocytes are somewhat similar, but smooth muscle is really a lot of things that contract smooth muscle. So, my favorite table is to give you an idea of the differences, because This is largely comparative. You have skeletal muscle, cardiac muscle, and smooth muscle. The excitation of skeletal muscle occurs at the neuromuscular junction is through a motor axon. Releases ACH, activates the thing, and that's how it's activated. Cardiac muscle is myogenic, which I'm going to hammer at you next week. And cells are coupled, electrically coupled through gap junctions, small cells. Smooth muscle, excited by synaptic transmission through the autonomic nervous system. Hormone receptors of various kinds will contract smooth muscle, or hormone, and they have hormone receptors. All sorts of things will actually activate calcium entry into smooth muscle. And some types of smooth muscle are coupled in sheets through gap junction. Calcium sensor, the big difference. Calcium sensor, the skeletal muscle is component C. Cardiac muscle, same guy, slightly different because it requires only one calcium ion. And in smooth muscle, calcium sensor is this protein called calmodulin. Excitation, contraction, coupling involves L-type calcium channels or anodine receptors. In cardiac muscle, you've got to have, this is probably mechanical or charge. No calcium has to enter the cell, and it usually doesn't require much. So no calcium involved here. In cardiac muscle, it's called calcium-induced calcium release. And you need calcium entry. Calcium is required. You cannot get smooth or cardiac muscle contract if the excitation or if there's no calcium outside. And if you want to uncouple in an experimental situation, in cardiac myocytes, you want to uncouple it, the contraction from excitation. If your electrophysiologists say, you could zero out the calcium and the muscle won't contract. Works very well, actually. So it requires calcium entry. In smooth muscle, the calcium entry is to multiple gated channels. These guys primarily IP3 modulated, and it also has randomine receptors. Termination of contraction, removal of calcium, ATP-dependent pump. It inhibits active myosin binding. Cardiac muscle is roughly the same, but sodium calcium exchange becomes a little more important, and same sort of mechanism. In smooth muscle, removal of calcium, ADP-dependent pumps, various kinds, and myosin light chain phosphatase. So that's the differences of the three types of muscle. We're going to come back to cardiac muscle and smooth muscle, cardiac. Any questions? All this is on the exam. I'm done. Done what I'm doing.